Here at the Bayer Research Centre in Toowoomba, we run a resistance monitoring program for the proteins in Bollgard 3 for Helicobacter species. What that involves is making sure that we're not seeing an increase in resistance alleles to the proteins in Bollgard 3 over time. Hello, I'm Dr Kristen Knight and I am the Insect Resistance Management Scientist at Bayer. We've been running this program since 2003, and what that involves is receiving samples which are eggs or Helicobacter larvae from all the cotton growing regions throughout Australia. Once we receive the samples, we rear them up to moths and we can do two screens, either the F1 screen or the F2 screen. At the end of that process, we will know if the individual from the field was carrying a resistance gene or not. At the end of the season, we collate the data, and what we're looking for is to make sure that the resistance allele frequency is not increasing over time. The data from the 21-22 season indicates that there's been no change or increase in resistance allele frequency to any of the proteins in Bollgard 3 for either of the Helicobacter species. These are really great results, and what it really reinforces is that the resistance management plan is working, and it's the work that farmers, new growers are doing on farm implementing that resistance management plan is ensuring that we are delaying the evolution of resistance to the proteins in Bollgard 3 over the many years that we've had this technology being grown in Australia. Stewardship of our insecticides and our traits are really important to those of us at Bayer, but they're also really important for the wider industry. Bollgard 3 is a genetically modified trait in cotton, and essentially it's acting as an insecticide all season long, and that's why we need to think about implementing both IPM and IRM in that system.